Welcome everyone to our lesson on muscle tissue. In this chapter, we'll explore one of the most dynamic and essential tissues in the human body. Muscle tissue allows us to move, pump blood, and digest food, even when we're not consciously thinking about it. We'll take a closer look at how muscle tissue is organized, how different types of muscle compare in structure and function, and how contraction allows movement. This is a foundational topic that helps us understand everything from lifting your arm to how your heart keeps beating. By the end of this chapter, you should be able to describe how muscle tissue is organized at the cellular and tissue level. You'll also need to compare and contrast the three types of muscle tissue: skeletal, cardiac, and smooth. We'll talk about how each type looks under the microscope, where it's found in the body, and what role it plays. Another goal is to understand how skeletal muscles pull on bones via tendons to generate movement. This involves learning about contraction and relaxation at the cellular level, an incredibly precise process. Finally, we'll touch on how muscle tissue develops during embryonic life and how well it can regenerate after injury. Keep these objectives in mind as we go through the material. They'll help guide your focus. Muscle tissue comes in three major types, each with unique features and functions. Skeletal muscle is the one most people think of. It attaches to bones and allows voluntary movements like walking or lifting objects. It's under conscious control and has a striated, striped appearance due to its internal structure. Cardiac muscle is only found in the heart. It contracts involuntarily and rhythmically to pump blood through the circulatory system. Like skeletal muscle, it is also striated, but it functions automatically without our input. Smooth muscle lines many of our internal organs, like the digestive tract and blood vessels. It's non-striated and completely involuntary, helping with processes such as digestion, blood flow regulation, and even making your hair stand up when you're cold. All muscle types work through contraction and relaxation, but the mechanisms and control differ. While we'll introduce all three, the main emphasis of this chapter will be on skeletal muscle, since it's the most complex and foundational to body movement. Before we dive into each muscle type, it's important to understand the four core functional properties that all muscle tissue shares, regardless of whether it's skeletal, cardiac, or smooth. The first property is excitability. This means muscle cells have the ability to respond to a stimulus, typically a signal from a motor neuron or the nervous system. When a stimulus is received, the plasma membrane of the muscle cell changes its electrical charge, generating an action potential. This wave-like electrical signal travels along the surface of the cell and initiates contraction. The second property is contractility, which is what most people associate with muscle tissue. This refers to the cell's ability to actively shorten. When a muscle contracts, it pulls on its attachment points, usually bones in the case of skeletal muscle, to create movement or tension. Cardiac and smooth muscles also contract, though in slightly different ways. The third property is extensibility. Unlike most tissues, muscle cells can be stretched without being damaged. This is critical during daily activities. For example, as one muscle contracts, the opposing muscle group must extend to allow that motion. Extensibility ensures that muscles can lengthen in response to movement or external forces. Lastly, elasticity is the ability of a muscle to return to its original shape after being stretched or contracted. This is possible thanks to structural proteins like titin, which act like molecular springs inside the muscle. Without elasticity, muscles would become slack or overstretched after every movement, making coordinated motion impossible. These four properties work together in harmony, allowing muscles to perform powerful, controlled, and repeatable actions, whether you're running a marathon or digesting a meal. Skeletal muscle is likely the type of muscle tissue you're most familiar with. It's what we use when we walk, lift, breathe, or even talk. These muscles are attached to bones via tendons and are under voluntary control, meaning we consciously decide when to activate them. Under the microscope, skeletal muscle fibers appear as long, cylindrical cells with a very clear striated pattern. These striations reflect the organized arrangement of actin and myosin proteins we discussed earlier. What's unique about skeletal muscle cells is that they are multinucleated. Each fiber contains multiple nuclei, which are located at the edges of the cell. This peripheral positioning leaves more space in the center of the fiber for contractile proteins. Skeletal muscle is incredibly massive in terms of contribution to body mass. It makes up roughly 40% of your total body weight and holds 50 to 75% of all body protein. That's because muscle tissue needs to be strong and densely packed with proteins to generate the force necessary for movement. These muscle fibers can be very long, some up to 42 centimeters or about 16.5 inches, such as those in the sartorius muscle, which runs along the thigh. Because of their length and volume, skeletal muscle fibers need multiple nuclei to manage cellular function across their entire structure. You'll find skeletal muscle in all the places that require voluntary movement. This includes the muscles that move your limbs and trunk, as well as less obvious areas like the muscles of facial expression, the diaphragm for breathing, and even sphincters like those controlling the anus and urethra. An interesting example is the tongue, which is composed of skeletal muscle too. Even though it's an internal structure, it's under voluntary control, and that's why we can articulate speech and move food during chewing. Understanding the structure and function of skeletal muscle provides a strong foundation for exploring how our bodies perform mechanical work and respond to neural control. Skeletal muscle is likely the type of muscle tissue you're most familiar with. It's what we use when we walk, lift, breathe, or even talk. These muscles are attached to bones via tendons and are under voluntary control, meaning we consciously decide when to activate them. Under the microscope, skeletal muscle fibers appear as long, cylindrical cells with a very clear striated pattern. These striations reflect the organized arrangement of actin and myosin proteins we discussed earlier. What's unique about skeletal muscle cells is that they are multinucleated. Each fiber contains multiple nuclei, which are located at the edges of the cell. This peripheral positioning leaves more space in the center of the fiber for contractile proteins. Skeletal muscle is incredibly massive in terms of contribution to body mass. It makes up roughly 40% of your total body weight and holds 50 to 75% of all body protein. That's because muscle tissue needs to be strong and densely packed with proteins to generate the force necessary for movement. These muscle fibers could be very long, some up to 42 centimeters or about 16.5 inches, such as those in the sartorius muscle, which runs along the thigh. Because of their length and volume, skeletal muscle fibers need multiple nuclei to manage cellular function across their entire structure. You'll find skeletal muscle in all the places that require voluntary movement. This includes the muscles that move your limbs and trunk, as well as less obvious areas like the muscles of facial expression, the diaphragm for breathing, and even sphincters like those controlling the anus and urethra. An interesting example is the tongue, which is composed of skeletal muscle too. Even though it's an internal structure, it's under voluntary control, and that's why we can articulate speech and move food during chewing. Understanding the structure and function of skeletal muscle provides a strong foundation for exploring how our bodies perform mechanical work and respond to neural control. Now let's focus on cardiac muscle, which is exclusively found in the heart. This muscle type powers the pumping action that moves blood through the circulatory system. Although it shares some characteristics with skeletal muscle, such as being striated, cardiac muscle operates involuntarily. You don't have to consciously think about making your heart beat. The cells of cardiac muscle, called cardiac myofibers, are shorter than skeletal muscle fibers and have a branched shape. Each fiber typically has one centrally located nucleus, although some may have two. A hallmark feature of cardiac muscle is the presence of intercalated discs, which are specialized junctions that connect one cardiac muscle cell to the next. These discs are clearly visible under the microscope and play two critical roles. Gap junctions allow electrical impulses, action potentials, to flow directly and rapidly from one cell to the next, enabling the synchronized contraction of the entire heart. Desmosomes anchor the cells together physically, ensuring that the muscle fibers remain connected even under the stress of powerful and repeated contractions. Cardiac muscle fibers are also packed with mitochondria and myoglobin, which supply the ATP and oxygen needed for sustained activity. Since the heart is constantly working, 24/7, year after year, It relies heavily on aerobic metabolism and needs to be fatigue-resistant. Under the microscope, cardiac muscle appears striated, but the striations are generally less prominent than in skeletal muscle. What stands out instead are the intercalated discs, which often look like dark, zigzagging lines between the fibers. Thanks to the combination of electrical coupling and strong physical connections, the heart contracts as a functional syncytium, meaning all cardiac fibers work as one unit. This is critical for effective blood pumping and maintaining a consistent rhythm. Overall, cardiac muscle is beautifully specialized for continuous, rhythmic, and automatic contraction, making it one of the most reliable and resilient muscle tissues in the body. Let's wrap up the muscle tissue types with smooth muscle, which has a very different appearance and function from the other two. You'll find smooth muscle throughout the body, especially in the walls of hollow organs and passageways, such as the digestive tract, urinary bladder, uterus, blood vessels, and even in the iris of the eye and skin. The cells of smooth muscle are shaped like spindles, wide in the middle and tapering at both ends. Each fiber contains a single nucleus located in the center of the cell. When viewed under the microscope, smooth muscle lacks visible striations. This is because the contractile proteins, actin and myosin, are arranged irregularly throughout the cytoplasm instead of in the neatly organized sarcomeres seen in skeletal and cardiac muscle. Despite lacking striations, smooth muscle is fully capable of contraction. In fact, smooth muscle is especially good at sustained contractions over long periods, often with minimal energy use. Contraction occurs when the actin and myosin pull on structures called dense bodies, which are linked to intermediate filaments inside the cell. This causes the entire fiber to twist or bulge in a corkscrew motion, pulling the ends of the cell inward. Unlike skeletal muscle, smooth muscle is involuntary, meaning you don't consciously control it. It's regulated by the autonomic nervous system, hormones, and sometimes local environmental conditions, like stretch or pH. This makes smooth muscle ideal for automatic functions like peristalsis in the intestines, controlling blood vessel diameter, or adjusting the shape of the eye's lens. Another interesting feature is that smooth muscle produces its own connective tissue known as the endomysium, which surrounds individual fibers and provides support and structure. Smooth muscle is versatile, resilient, and found almost everywhere in the body. Although it doesn't grab attention like skeletal or cardiac muscle, it plays a vital role in regulating internal processes without us even being aware of it. This final slide brings everything together in a single comparison table to help you see the similarities and differences between the three muscle types at a glance. This is especially helpful for lab identification and when you're trying to remember which type of muscle tissue performs which functions in the body. Let's start with skeletal muscle. It's voluntary, striated, and composed of long cylindrical cells with multiple nuclei. These cells are designed for quick, forceful contractions to move the skeleton and body parts. Cardiac muscle is also striated, but it's involuntary and has short branched fibers that are connected by intercalated discs. These specialized junctions allow the heart to beat in a coordinated, rhythmic manner. Cardiac muscle has one or sometimes two nuclei, and it's rich in mitochondria for continuous activity. Smooth muscle, on the other hand, is non-striated and composed of spindle-shaped fibers with a single central nucleus. It contracts slowly, but can sustain contractions longer, making it ideal for internal organs that need continuous, low-level activity, like the intestines or blood vessels. Structurally and functionally, each muscle type is specialized for the job it performs in the body. Skeletal muscle handles fast voluntary movements. Cardiac muscle coordinates powerful, rhythmic contractions of the heart. Smooth muscle handles involuntary movements, essential for life, like digestion and blood flow. Understanding these differences will also help you during lab practicals, especially when viewing histology slides. So be sure to review this table regularly. Skeletal muscle is most well known for its role in producing movement. When you walk, lift your arm, blink, or even type on a keyboard, skeletal muscles are contracting in a coordinated way to pull on bones and generate motion. This movement is under voluntary control, meaning you choose when to engage these muscles, unlike cardiac or smooth muscle. But movement is only part of what skeletal muscles do. A major yet often overlooked function is resisting the force of gravity to maintain posture. Whether you're standing, sitting, or balancing on one foot, your muscles are constantly making tiny adjustments to keep you upright and stable. These subtle contractions happen continuously, even when you're not consciously aware of them. Another key function is joint stabilization. Muscles surrounding a joint help keep it aligned and prevent it from being pulled too far out of its natural range of motion. This is particularly important for joints that bear weight or are frequently used, such as the shoulder, knee, and spine. By stabilizing joints, skeletal muscles prevent injuries, such as sprains or dislocations. Skeletal muscles also play a vital role in regulating openings of internal tracts. For example, muscles in the throat control swallowing. Muscles in the urethra and anus allow voluntary control over urination and defecation. These are crucial not only for function, but for social and developmental milestones like potty training. Additionally, skeletal muscle serves a protective role. In the abdominal and pelvic regions, muscle layers act as a shield, supporting and defending internal organs like the intestines, bladder, and reproductive structures. Think of your abdominal wall. Those muscle layers absorb impact and help prevent injury from trauma. Finally, skeletal muscles are important for thermoregulation. When muscles contract, they use ATP, and a byproduct of this process is heat. During exercise, your muscles produce significant heat, which increases your core body temperature. In cold environments, shivering, a reflex of rapid skeletal muscle contractions, helps maintain a stable internal temperature by generating heat involuntarily. This function helps preserve homeostasis, which is the body's ability to maintain stable internal conditions. So while movement is the most obvious function of skeletal muscle, it is just one piece of a much larger picture. Skeletal muscles contribute to stability, protection, control, and even survival through heat production. A single skeletal muscle such as your biceps brachii or quadriceps femoris is not just one type of tissue. It's considered an organ because it consists of multiple tissue types working together. Skeletal muscle fibers, connective tissue layers, blood vessels, and nerves. All of these components are critical for muscle function, coordination, and survival. One of the most important structural features of skeletal muscle is its connective tissue framework, known collectively as the mesia. These layers wrap around different levels of the muscle and provide both support and organization. The epimysium is the outermost layer. It surrounds the entire muscle organ and is made of dense, irregular connective tissue. This tough layer gives the muscle its shape and strength, allowing it to contract without falling apart. It also separates the muscle from surrounding tissues and organs, helping it move independently. Inside the muscle, fibers are grouped into visible bundles called fascicles. Each fascicle is wrapped in the perimysium, another layer of dense, irregular connective tissue. Fascicles are like subdivisions within the muscle that allow for fine-tuned control. The nervous system can activate specific fascicles for partial or graded muscle contraction. Within each fascicle are individual muscle cells, or myofibers, and each is wrapped in the endomysium, a thin layer of areolar connective tissue. This layer houses capillaries and nerves and also surrounds each fiber with extracellular fluid and nutrients needed to keep the cell alive and functional. These three layers, epimysium, perimysium, and endomysium, also work together to transmit the force generated by muscle contraction. When muscle fibers contract, they pull on the endomysium, which transmits that force to the perimysium, then to the epimysium, and ultimately to the tendon, which pulls on the bone to produce movement. The compartmentalization provided by these layers is important for muscle repair and regeneration as well. If a portion of a muscle is damaged, the surrounding connective tissue can limit the spread of injury and inflammation. It's also important to note that nerves and blood vessels must reach every muscle fiber for the tissue to function. These travel through the connective tissue layers, entering with the epimysium and branching inward to reach each fascicle and individual myofiber. So while skeletal muscle is known for its contractile abilities, its organization as a complex, integrated organ is what makes those contractions precise, powerful, and sustainable. Skeletal muscle wouldn't be able to move the skeleton if it weren't for its attachments. And this is where tendons and aponeuroses come in. Tendons are tough bands of dense, regular connective tissue that serve as the anchor points between muscles and bones. They are the structures that physically transfer the force generated by muscle contraction to the skeleton, resulting in movement. When you flex your biceps, for example, the tendon pulls on your radius to bend your elbow. These tendons are not separate from the muscle. They are actually formed by the fusion of all three mysia layers, the endomysium, paramysium, and epimysium. As these connective tissues extend beyond the muscle fibers, they bundle together and form the cord-like tendon, or, in some locations, a broad aponeurosis. An aponeurosis is essentially a flat, sheet-like tendon used when a muscle needs to anchor over a broader area, or when multiple muscles share a common insertion. A great example is in the lower back, where the latissimus dorsi muscles converge into a broad aponeurosis. At the attachment point, the collagen fibers of the tendon blend into the periosteum, the dense connective tissue that surrounds the bone. This creates a strong, continuous link from muscle to bone, allowing force to be effectively transmitted during movement. Now, for muscles to contract and do their work, they need a rich blood supply. Every skeletal muscle is highly vascularized to deliver oxygen and nutrients, and to remove waste products like carbon dioxide and lactic acid. Capillaries run through the endomysium to supply individual muscle fibers. Lastly, each and every muscle fiber is directly connected to a motor neuron through a structure called the neuromuscular junction, which we'll cover more in a later slide. These neurons are the only way skeletal muscle can be activated, unlike cardiac or smooth muscle, which can self-regulate to some degree. Skeletal muscle requires neural input to contract. Together, the connective tissue layers, tendons or aponeuroses, blood supply, and nerve supply ensure that skeletal muscle is structurally integrated, metabolically supported, and functionally controlled. Let's take a closer look at the individual cells of skeletal muscle, which are called muscle fibers or myofibers. These are not typical cells, they're long, cylindrical, and multinucleated, often reaching up to 30 centimeters, almost 12 inches, in length. You can actually see these fibers running along the direction of the muscle in a good dissection or histological slide. How do these massive cells form? During embryonic development, myoblasts, which are individual single-nucleated precursor cells, fuse together to form one large syncytial cell. This process is unique to skeletal muscle and results in a fiber with multiple nuclei, which are typically located at the periphery of the cell, just beneath the sarcolemma. Why so many nuclei? Skeletal myofibers need to produce huge amounts of contractile proteins to maintain and repair themselves and to sustain muscle contractions. Multiple nuclei allow the fiber to efficiently produce and regulate proteins and enzymes along its entire length. Inside each myofiber are numerous myofibrils. These are rod-like structures that run parallel to the long axis of the fiber. Each myofibril is made up of repeating segments of contractile proteins, primarily actin, thin filaments, and myosin, thick filaments. These filaments are arranged in highly organized, repeating patterns called sarcomeres, which are the functional units of contraction. We'll cover them next in detail. There can be hundreds to thousands of myofibrils within a single myofiber. Despite their small diameter, around to the 1.2 micrometers, together they give the muscle fiber its strength and contractile capacity. When the myofibrils contract, the entire cell shortens, because they are attached to the sarcolemma at each end of the fiber. Surrounding the myofibrils is the sarcoplasm, which is the specialized cytoplasm of the muscle fiber. It contains glycogen, myoglobin, and various enzymes needed for energy production. The outer membrane of the fiber is called the sarcolemma. This is the structure that conducts the electrical signals, action potentials, along the muscle surface to initiate contraction. The level of organization from fiber to myofibril to myofilament shows just how precise and specialized skeletal muscle tissue is. This structural complexity allows muscles to contract in a coordinated, powerful, and controlled manner, something essential for voluntary movement. The sarcomere is the most fundamental structure of muscle contraction. It's often referred to as the functional unit of the skeletal muscle fiber. A sarcomere extends from one Z-line to the next Z-line, and there are thousands of them arranged end-to-end along the length of each myofibril. Within a sarcomere are two main types of myofilaments: thin filaments, which are primarily composed of actin, along with two regulatory proteins, troponin and tropomyosin. Troponin binds calcium and controls the position of tropomyosin, which blocks or exposes the myosin binding sites on actin. Thick filaments, which are composed of myosin molecules. Each myosin has a tail region and a pair of heads that can bind to actin and to ATP, which fuels contraction. When you view skeletal muscle under a microscope, the striations you see are the result of the precise alignment of these filaments. Specifically, the A band is the dark band, where thick filaments, myosin, are located. In the middle of the A band is the H zone, which contains myosin only, no actin overlap. The I band is the light band, where only thin filaments, actin, are found. The M line runs down the center of the sarcomere, holding the thick filaments in place. The Z line marks the boundary of each sarcomere and anchors the actin filaments. During contraction, sarcomeres shorten, but it's important to emphasize that the filaments themselves do not change length. Instead, they slide past each other, a process known as the sliding filament model. As myosin heads bind to actin and pull, the Z lines are drawn closer together, the I bands narrow, and the zone of overlap increases. This collective shortening of thousands of sarcomeres causes the entire muscle fiber, and eventually the whole muscle, to contract. When the muscle relaxes, the filaments slide back to their original positions, and the sarcomere returns to its resting length. This highly organized arrangement ensures a consistent and efficient contraction mechanism across all fibers in the muscle. Understanding sarcomere structure is essential for grasping how force is generated at the microscopic level and how it translates to visible movement at the macroscopic level. Now that we understand the structure of the sarcomere, let's look at how muscle contraction is actually initiated. The process begins with a signal from the nervous system, specifically a somatic motor neuron. The point where this motor neuron connects with a skeletal muscle fiber is called the neuromuscular junction, or NMJ. The neuron's axon terminal sits very close to the muscle fiber's membrane, but doesn't physically touch it. There's a tiny gap called the synaptic cleft. When a nerve impulse, or action potential, reaches the axon terminal, it triggers the release of a neurotransmitter called acetylcholine, ACH, into the synaptic cleft. ACH diffuses across this space and binds to specialized ACH receptors located on the muscle fiber's membrane in a region called the motor end plate. This binding causes an electrical change in the sarcolemma, generating a new action potential in the muscle fiber. That electrical impulse travels along the sarcolemma and dives deep into the fiber via structures called T-tubules, which are invaginations of the membrane that reach into the fiber's interior. Surrounding each myofibril is a network called the sarcoplasmic reticulum, SR, which functions like a storage tank for calcium ions, Ca² mice. The action potential reaching the T-tubules stimulates the SR, specifically the terminal cisternae, to release calcium into the sarcoplasm. Calcium plays a critical role of initiating contraction. It binds to the regulatory protein troponin, which then causes a conformational change that moves tropomyosin away from the myosin binding sites on the actin filament. Once those sites are exposed, the myosin heads can bind to actin, and the contraction cycle begins. This is known as the sliding filament mechanism. As myosin pulls on actin, the sarcomere shortens, and this collective shortening across all sarcomeres in a fiber causes the entire muscle to contract. The process continues as long as calcium remains available and ACH continues to signal the fiber. Once the nerve signal stops, ACH is broken down, calcium is pumped back into the SR, and the muscle fiber relaxes. This precise and coordinated system ensures that skeletal muscle contraction is both voluntary and controllable. This slide brings everything together into one cohesive overview of skeletal muscle structure and function. First, recall the diverse roles of skeletal muscle. It's not just about movement. Muscles also help you stay upright, stabilize joints, regulate openings, protect organs, and maintain body temperature, especially when contracting generates heat. At the structural level, skeletal muscle is a complex organ system. It consists of specialized muscle fibers wrapped in 3 layers of connective tissue: epimysium, paramysium, and endomysium. These layers help transmit the force of contraction and keep everything organized and supported. These connective tissues come together to form tendons, cord-like, or aponeurosis, sheet-like, that attach muscles to bones or fascia. This structural continuity allows for efficient transfer of force to the skeleton during movement. Inside each myofiber, myofibrils run lengthwise and house the contractile machinery, repeating units of sarcomeres. Sarcomeres are made of thin actin and thick myosin filaments, which slide past one another to shorten the muscle. This is the microscopic basis for the striated appearance of skeletal muscle and the core mechanism of contraction. Finally, muscle contraction is initiated by the nervous system. A signal from a motor neuron reaches the muscle at the neuromuscular junction, triggering the release of acetylcholine, ACH. This leads to an electrical signal in the muscle fiber, release of calcium ions from the sarcoplasmic reticulum, and activation of the sliding filament mechanism. Once contraction is complete, calcium is reabsorbed and the muscle relaxes. Together, these elements explain how skeletal muscle functions at every level, from whole body movement to microscopic protein interactions. Understanding this system gives you insight into everything from athletic performance to neuromuscular diseases. To understand how skeletal muscles create movement, it's important to recognize that they don't push, they only pull. When a muscle contracts, it generates tension, which is transferred to a tendon. Tendons are rope-like structures made of dense, regular connective tissue, specifically rich in collagen. These structures are extremely strong and designed to withstand tension, which allows them to function like cables, pulling bones during movement. Tendons connect directly into the periosteum, the tough connective tissue that covers bone surfaces. This secure integration ensures that when the muscle shortens, the force is directly transferred to the bone to create movement at a joint. Each muscle has two main attachment points. The origin is the stationary or fixed point. It usually lies proximal, closer to the trunk of the body. The insertion is the movable point. It typically lies distal and is pulled toward the origin during contraction. For example, when you flex your elbow, the biceps brachii contracts, pulling the radius, insertion, toward the scapula, origin, bending the arm. However, not all skeletal muscles attach to bones. Some control movements of the skin, like facial expressions, or function as sphincters to regulate the opening and closing of passageways, such as the mouth, anus, or urethra. Others, like the diaphragm, act on body cavities to help with breathing rather than bone movement. Understanding the layout of origin and insertion also allows us to predict direction of movement, which is critical when analyzing body mechanics or identifying muscle function in dissection and imaging. So whether you're lifting your arm, puckering your lips, or exhaling forcefully, skeletal muscles transmit force through their attachments to perform vital functions across the body. Skeletal muscles work together in a coordinated system to create smooth and controlled movements. Rather than acting alone, muscles function as part of a team with different roles depending on the motion being performed. The main muscle that initiates and performs a specific movement is called the agonist or prime mover. For example, during elbow flexion, such as when lifting a mug, the biceps brachii is the agonist. It's the muscle generating most of the force for that action. On the flip side, we have the antagonist, which produces the opposite movement. In that same example, the triceps brachii acts as the antagonist by resisting the motion of the biceps and helping to control the speed and precision of the movement. This role is crucial, not just to oppose the agonist, but to stabilize joints and prevent overshooting or injury. Then there are synergists, which support the agonist in its function. Synergists can add strength, reduce unwanted motion, or help fine-tune movement. For example, the brachialis and brachioradialis both assist the biceps brachii during elbow flexion. Without them, the movement would be less stable and possibly less efficient. A special type of synergist is the fixator. A fixator helps by anchoring the origin of the agonist so that the contraction has a firm base from which to pull. In our earlier example, the muscles that stabilize the scapula serve as fixators during arm movement, allowing muscles like the biceps to do their job without wobble at the shoulder. One of the most important takeaways here is that muscle roles are dynamic. Depending on the action, a muscle that acts as an agonist in one movement may become an antagonist in the opposite movement. For example, the quadriceps femoris group extends the knee, agonist in extension, but during flexion, the hamstrings become the agonists and the quadriceps become the antagonists. This dynamic interplay allows us to perform highly controlled, adaptable, and precise movements, whether we're lifting a pen or performing a high-speed athletic action. In clinical terms, understanding these interactions helps in evaluating muscle injuries, strength training, physical therapy regimens, and even surgical planning. Disrupting any one component of this coordinated team can lead to dysfunction or instability. In skeletal muscle anatomy, how the muscle fibers are organized, called fascicle arrangement, plays a major role in determining how a muscle functions. Fascicles are groups of muscle fibers bundled together by perimysium, and their alignment impacts both how much force the muscle can generate and how far it can contract. Let's look at the major fascicle patterns. Parallel muscles have fibers that run in the same direction as the muscle's long axis. These muscles contract over a long distance and have good range of motion, but they don't generate as much force. A classic example is the sartorius in the thigh. A subtype of parallel muscle is fusiform, which has a thick belly and tapers at both ends, like the biceps brachii. Fusiform muscles can still contract over a good range, but generate slightly more force due to their shape. Circular muscles, also known as sphincters, have fascicles arranged in concentric rings. These control openings, think of muscles around the mouth, orbicularis oris, or around the eyes, orbicularis oculi. When they contract, they close the opening. When they relax, they allow it to open. Convergent muscles have a broad origin and narrow down to a single point of insertion, like a triangle. This arrangement allows versatile movement because different regions of the muscle can be activated independently. The pectoralis major is a prime example. It spreads out over the chest and narrows into the tendon attaching to the humerus. Then we have pennate muscles, which are arranged like a feather, with fibers attaching at an angle to a central tendon. This reduces range of motion, but greatly increases force output, because more fibers can be packed into the same space. This classification is not just for anatomy exams. It has real-world applications in understanding movement efficiency, muscle injury patterns, and even athletic performance. For example, sprinters often train pennate muscles for explosive force, while dancers and yogis focus on flexibility in longer, parallel muscles. Recognizing these arrangements helps you predict function from form, an essential skill in anatomy and clinical practice. One of the most fascinating aspects of muscle function is how skeletal muscles and bones act together as lever systems, just like machines. A lever is a rigid bar, in this case a bone, that moves on a fixed point called a fulcrum, which is typically a joint. When a muscle contracts, it produces a pulling force known as the effort. This effort is applied at a specific point on the lever, bone, and it moves a load that could be a body part, an external object, or simply the force of gravity acting on the body. These lever systems are classified based on the relative positions of the fulcrum, F, effort, E, and load or resistance, R. A first-class lever has the fulcrum between the load and the effort. A real-world example is a seesaw. In the body, this occurs during neck extension. The atlanto-occipital joint serves as the fulcrum. The weight of the head is the load, and the neck muscles, e.g. trapezius, provide the effort on the other side. A second-class lever has the load between the fulcrum and the effort. Think of a wheelbarrow. In the body, this happens when you stand on tiptoe. The ball of the foot is the fulcrum, the body weight is the load, and the gastrocnemius muscle in the calf provides the effort. This type increases force but sacrifices speed and distance. A third-class lever has the effort between the fulcrum and the load, like a pair of tongs. This is the most common lever system in the human body. An example is elbow flexion. The elbow joint is the fulcrum. The biceps brachii applies effort on the forearm, and the load is whatever you're holding in your hand. These levers are designed for speed and range of motion, not power. Lever systems help us understand why certain muscles are located where they are, and why some movements are fast but weak, while others are strong but limited in range. For example, even though the biceps don't generate as much force as your quadriceps, the placement of the fulcrum and effort allows rapid arm movements, ideal for actions like throwing or lifting light objects quickly. It connects anatomy with physics, showing how form and function are perfectly matched in the human body. Many students feel overwhelmed by the sheer number of muscles to memorize. But what makes this process easier is understanding why muscles are named the way they are. Our journey in muscle naming begins with the ancient Greeks and Romans, who were among the first in Western history to systematically study the human body. As anatomical science evolved, later scholars retained Latin and Greek terms because these languages were used by the educated elite and early scientists. That tradition stuck, and now most anatomical terms, including the names of muscles, still use these classical languages. That's why so many names sound unfamiliar at first. They aren't English words, but once you learn their roots, they start making logical sense. This is where etymology becomes important. Etymology is the study of the origin and evolution of words. By learning the roots, like ab, meaning away from, or rectus, meaning straight, you can interpret a muscle's name without just memorizing it blindly. For instance, abductor digiti minimi literally translates to the muscle that moves the smallest finger away from the midline. Once you understand that, it's easier to remember its location and function. So think of muscle names as built-in descriptions. If you take the time to learn the building blocks, those Latin and Greek roots, you gain a powerful tool for success in anatomy. It's not just about memorization. It's about building a language of the body. On this slide, we're diving deeper into how muscle names are structured using Latin roots. Once you start recognizing these patterns, the names become a helpful description tool, not just a label. First, many muscles are named for their action. For example, a flexor bends a joint, while an extensor straightens it. An abductor moves a limb away from the body's midline, while an adductor brings it closer. Next, we can identify a muscle's location from its name. For instance, brachii tells us it's in the arm, brachial region. Femoris relates to the femur in the thigh, and frontalis lies over the frontal bone. Size and length are often built into the name too. Maximus, largest, minimus, smallest, longus, long, and brevis, short. Knowing these helps you differentiate between muscles in a group, like the gluteus maximus, medius, and minimus. Shape also plays a role. The deltoid is named for its triangular, delta-shaped form. Orbicularis means ring-shaped or circular, as in the muscles around the mouth and eyes. Fiber direction gives more clues. Rectus means straight, like the rectus abdominis. And oblique means angled, describing the oblique muscles. Some muscles are named by how many origins they have. Biceps has 2, triceps has 3, and quadriceps has 4. So as you can see, each muscle name is like a code. If you can decode the Latin or Greek roots, you gain a clearer understanding of where a muscle is, what it does, and how it looks. This not only helps with memorization, but it deepens your anatomical insight. On this slide, we're organizing the most common ways skeletal muscles get their names. Understanding these naming criteria helps demystify anatomy and gives you a logical way to learn hundreds of muscles more efficiently. First, muscles are often named for their location. For example, the frontalis is on top of the frontal bone of the skull. And the tibialis anterior lies on the front of the tibia. Shape is another major clue. Muscles like the deltoid are triangular, and the trapezius forms a trapezoid shape across the upper back. Size comparisons are also common. Think of the gluteus muscles. Maximus is the largest, medius is intermediate, and minimus is the smallest. Then there's fiber direction. If the fibers run straight, the muscle is usually called rectus. If they run diagonally, it's oblique. And if horizontal, transversus. This is especially helpful when studying abdominal muscles. Muscles are also named by how many origins they have. The prefix bi means 2, like biceps brachii. Tri means 3, triceps brachii, and quad means 4, quadriceps femoris. Some muscles are named for their attachments, and in these cases, the name follows a pattern from origin to insertion. A perfect example is the sternocleidomastoid, which originates on the sternum, sterno, and clavicle, clado, and inserts on the mastoid process. Finally, some muscle names describe their action. This is where you'll see terms like flexor, extensor, abductor, or adductor, each telling you how the muscle moves a part of the body. Once you get comfortable with these patterns, the names stop being arbitrary. They actually tell a story about each muscle's structure and function. In this final slide of the naming section, I want to emphasize why all this terminology matters. Beyond just passing a test or labeling diagrams, learning the etymology, the origin and meaning of words, helps you make sense of long, complex muscle names. For example, the name abductor pollicis longus may sound intimidating, but once you break it down, it simply means a long muscle that abducts the thumb. Pollicis equals thumb. These names are not arbitrary. They're descriptive tools that help you understand where a muscle is, what it does, and how it connects to other structures. In that way, they mirror the anatomy itself. This knowledge is incredibly useful in clinical settings too. Whether it's physical therapy, surgery, or diagnostics, health professionals use this terminology daily. For example, if someone injures their flexor carpi radialis, you can predict where the pain is and what function is affected. Another advantage is that Latin names provide a universal scientific language. Whether you're reading a medical chart in English, Spanish, or French, terms like gluteus maximus and rectus abdominis are understood the same way. This standardization reduces confusion in global healthcare and scientific communication. Finally, there's the personal benefit. When students begin to decode these names, they feel more confident and empowered. What seems like an overwhelming list of terms becomes a series of logical, memorable patterns. And that shift in mindset makes all the difference as we continue exploring the muscular system.